screen?
Yeah, sure.
Yes.
Yeah. All right. Perfect. Okay, so we can get started. So Seb and I will be presenting today together, and we came here to present our work on virus shaping, and that is opponent shaping applied for antibody development, specifically to develop, or potentially in the future, develop antiviral therapies that are better than the current ones that we have. And we are both students at the University of Oxford in the UK. We are part of the same lab. We have the same supervisor, Jacob Forster. And yeah, we're like more or less midway through our PhDs. I don't know if you want to say anything extra.
Oh yes, I'm also co-supervised by Harrison Steele, who is also helpful because he's a more of a engineering synthetic biologist, so he's helpful for the biology end as well.
Yeah. And I also have the co-supervisor who's from a previous company that I've worked for that's called Exentia, and she's a structure bioinformatician. So both of us have Sab has a computer science and maths background. I have a computer science background. So the biology stuff we've been kind of learning along the way. I picked bits and pieces when I was working at my previous company. But yeah, it's something that we're both interested in. And hence why this project. By the way, if you have any questions along the way, please feel free to just raise your hand or unmute yourself and ask the question. Like the more interactive this is, the nicer I think for everyone. So don't be shy and jump in. You don't need to wait until the end.
Thank you, Alexander, for that.
Yeah, of course.
Yeah.
So today we came to you with a little presentation of two stories. One is the story of our actual project. And the motivation behind our project is that currently when we design therapies, the way they're designed is in a myopic sense. So we think about the current pathogen that's out there. So that can be something like COVID, for example. We try to design therapies that would work with the current variant of, let's say, COVID. But in case of viruses we know are adaptive. They're adaptive agents. So again, if you think back to COVID and when the COVID vaccine was released, for example, the COVID vaccine was released, but then soon after we realized that there's going to be new variants emerging. And then as the new variants were emerging and people were testing the efficacy of the vaccine against the new emerging variants, they realized that, oh crap, like our therapies might not work so well with the new variants. So that's a problem. And that's something that we wanted to address in this project. And then we realized that we can sort of take advantage of the fact that the viruses adapt in response to the therapies that we make. So we can use this and furthermore sort of like make this part of like an agency that we have over the virus, because we know whatever we do as the sort of like mastermind behind the therapy, the viruses will respond directly to that thing. So in the design process already, we can take that into account and take advantage of that little bit of agency that we have over the viruses. So that's something that we'll talk about a little bit more throughout this presentation. But that was the motivation behind the project. So we want to design therapies against viruses that are not myopic and that take advantage of the agency that we have over viruses. And then there's the other story, which is a little bit more humorous, that we hope to tell you about a little bit, which is when we first started working on this project and we went to present it at like a small conference. We were making a poster for it, and Seb generated this great picture of DALL-E that you saw on the first slide, where there was a virus and the antibody, and we thought it was very cute. We loved it, and then we went and we showed it to our supervisor, and he didn't like it. And he was like, well, this picture is not a good representation of what actually happens here, because it shows that the antibody and the virus are sort of like playing the same game, so he didn't like it. So we're like, well, okay, how about to show that the viruses are myopic, we'll add a blindfold on top of the virus, so the virus can actually see the chessboard in front of it. And then he was like, okay, fine. So then we tried to generate, we wanted the same image just with the added blindfold, and then we would prompt. Well, at that point it was already ChatGPT. We would prompt ChatGPT with the same image, and we were like, just change as little as possible, add a blindfold to the virus. So you'll see how that goes as we present. Okay, so first of all, we want to talk a little bit about the terms that we'll be using throughout the presentation. So there's the antibody, which in our case, they're the good guys. So that they are the thing that we control. They will be, in our work, they're synonymous to therapy. Basically, the antibody is the thing that we're designing. And then in actual world, antibodies are part of your immune system, and they're the thing that helps you fight off a bunch of different pathogens. But people also develop antibodies as a therapy against certain diseases. So for example, as a cancer drug, antibody therapeutics are actually, like, I think, the most common type of therapy. In case of viruses, often we talk about vaccines. Vaccines are not the same as antibodies. There's a step between a vaccine and the antibodies that your body would start producing. But for the purpose of this project, we're going to assume that we have a control over the antibody. And antibody will mean the therapy, and antibody is the thing that we're optimizing. And then there's the other thing, which is the virus. And specifically, what we look at is a viral antigen. Antigen is a small part of the virus that is actually the thing that will trigger the immune system to start its response. But in antigens and antibodies, they interact together. But if we would keep saying antibody and antigen all over, these terms are kind of confusing, and they sound very similar. So even though we mean antigen, for the rest of this presentation, we will just keep saying virus. But for any biology-minded folks out there, we mean antigen. All right, and then the key difference that is sort of like the foundation of this work that we did is that we want the antibodies, so the therapies, the thing that we design, we want to sort of like use the fact that humans can be sometimes a little bit smart, and we can actually realize that the virus will adapt, and we can account, when we're designing these antibodies, we can account for the viral adaptation. And furthermore, going back to this point of the agency over the virus, we can try to influence how the virus will adapt in response to the antibodies that we've designed. This is in contrast to the virus, which is a naive agent. Like, the virus can only ever realize what's like there at the minute. It's gonna like sort of like respond to the evolutionary pressures that are out there at the moment, but it has no foresight about what's going to happen. So we will model it as if it responds or attempts to respond optimally at any given point, but only to what's happening at the minute, not so like the virus will always play sort of myopic best response. All right, so with that, the goal of our work was to design this type of pipeline. So if you start at the top of the left loop, there's a starting antibody, and this is, in our case, a random antibody. And then we generate new possible antibodies. We do that by like applying random mutations. And then the key thing is that when we're going to be evaluating the performance of the antibody, we will then account for possible viral evolution. So then we enter this like viral evolution box, and from the starting virus, we generate the possible viruses, and we will evaluate them against the current antibody that's under evaluation. And we will evaluate the strength of the binding, so the strength of the interaction between the virus and the antibody. And for the virus, we're going to say that all the virus cares about is escaping from the antibody. So the virus doesn't want a good immune response. It wants to like just spread and do the viral things. So it's going to try to escape from the antibody. So for the virus, it'll try to minimize the strength of this interaction between the antibody and the virus. And then based on this binding strength and where it achieves the minimal strength, we're going to select the best viruses and repeat this loop. And we will repeat this for the horizon number of steps. So that's a pretty key term because we'll then refer to different antibodies optimized with different horizons as like shapers of horizon n. And then we come back to the antibody. So now that we've simulated this viral evolution for the horizon number of steps, we would have repeated it a few times. We'll talk about this a little bit more in a second, but then we come back to the antibody and then we select the best antibodies with respect to this more like performance that's averaged over the whole possible viral evolution. And then we pick the next antibody and we keep optimizing it for a fixed number of steps. So that's our goal. That's what we want to achieve. And for any more machine learning minded people in the audience, this is very similar to meta-learning. So if you think about this loop as meta-learning, the antibody optimization loop, that would be the outer loop in your optimization, and then the viral escape, that would be the inner loop. And as with any meta-learning approach, there's a problem. And the problem
Alexander, a question from me.
Yes.
In the process of meta learning and optimizing the antibody, I probably have missed what motivates the improvement on the antibody in order to put it back into this loop.
All right. Yeah. So the antibody will be optimizing. We'll talk about this a little bit more in a second, but the antibody will be optimizing for binding as well as it can to the virus, with the caveat, and I'll describe this a little bit more in a few slides, with the caveat that the antibody doesn't just want to bind to the current virus, it will care about binding to the viruses over the whole possible evolutionary trajectory of the virus. But the antibody basically has the opposite objective to the virus. So the virus cares about a weak interaction between the virus and the antibody, and the antibody wants a strong interaction between the virus and the antibody, so strong binding.
So I would say like the optimization is done such that we have stronger binding to the antigen.
Yes, that's right.
Okay, thank you.
But yes, but very importantly, across the whole thing as well. Like the virus can only ever look one step ahead, whilst if you repeat this loop 100 times, the antibody looks at all 100 steps of viral evolution.
Yeah, oh, thank you. Makes more sense.
Okay, cool. Sorry, I should have said that. So thank you for the question. And yeah, but back to my point about meta-learning. Meta-learning tends to be quite, it tends to require a lot of samples. So we realized that there's a problem, and that is that this binding evaluation that we need both for the viral fitness and for the antibody fitness, the speed of how fast we can calculate this binding strength, that's going to matter a lot for the feasibility of this method. So we needed this thing in the box to be very, very fast. And that was the first problem that we've encountered. Because not only did it need to be fast, but because we will be in our, the method that we've just described, we're changing both the virus in the inner loop as we simulate the escape and the antibody as we keep optimizing it. So we needed the binding simulator to be fast and to allow us to work with not only a specific virus, which is what a lot of different binding models can do, but we needed a model that would allow us to mutate the virus and still give us like reasonable binding scores. So that was the first crux that we've encountered. And then after chatting to a bunch of people, we learned about this model that's called Absolut. And that was really good for our purposes because it allowed us to basically work with mutations in the virus and mutations in the antibody. And the model still stayed relevant. And it was also already reasonably fast. So we were very happy when we made this discovery. However, the absolute binding framework comes at a cost of not being extremely accurate. So it's a massively simplified model of reality. But we figured that because this is more of a proof of concept type of project where we just want to show that this type of approach is feasible and it can yield, you know, like interesting results, this was good enough. So we decided to go with Absolut, and not only that, but we also decided that we can probably make it even faster. And Seb actually re-implemented the binding computation from Absolut in this framework that's called JAX, which is very good for parallelized GPU accelerated computation. So Absolut before was all the calculations would happen just on a CPU, but with the JAX re-implementation, it was now GPU accelerated and easily parallelizable. So that's how we got a big time speed up, which was like of an order of 10 to the -4. So now we had to fix our second problem. The binding simulator was sort of like solved, but then we had to think about how we're actually going to set up the incentives, the optimization incentives for the antibody and for the virus. So the main thing, like we already talked about before, the main thing for the antibody that it cares about is that it wants to bind to the virus. So that's what the green arrow indicates. However, for the virus, it wants to not bind to the antibody. So they have opposing objectives. But if we just modeled that in our model, then there's a very trivial solution that they would both collapse into. And that is that the virus can just become very non-sticky, so not bind to not just our antibody, but anything. And then the antibody can become very sticky, so bind to literally anything, which is extremely unrealistic. Like they would be a very bad antibody and a very bad virus. And it's also like from the perspective of this analysis, completely uninteresting as well. So we had to fix that. So we came up with this virus antibody game where we've added, for the antibody, we've added an anti-target. So a thing that the antibody can't bind to, or we don't want it to bind to. And then for the virus, we've added a target. So something that the virus should bind to. And if you think about, if you would want sort of like equivalence of this in reality, so how this sort of like anchors us into a more realistic setting, the viral target would be something like a cell receptor that the virus needs to interact with to actually enter the host cell and replicate. And then the anti-target would correspond, for the antibody, would correspond to something like a protein that just occurs in our bodies. So let it be like a receptor of a lung cell or something. You don't want your own immune system to attack your lungs. That would be very bad. So that would be like an equivalent of an anti-target. So we fixed these incentives where the antibody could no longer just become sticky and the virus could no longer just become non-sticky by adding these targets and anti-targets. And we've modeled our game as a two-player zero-sum game. So that means that the objective, the payoff for the antibody and the payoff for the virus were negatives of each other. And so now we are ready for this whole setup. So if we were to formalize what the antibody is actually optimizing, so coming back to your question from earlier, it is optimizing for its own payoff, which would be, so if you look at this line at the bottom here, this is bind to the virus, don't bind to the anti-target, and it also indirectly optimizes for the antibody wants the virus to not bind to its target indirectly. So that's the immediate payoff, the sort of like the myopic objective. But we want to model this long-term behavior against evolving viruses. So we're going to care about this payoff, but averaged over the escape trajectory of the virus. So that's the first expectation. And then averaged over many possible viral trajectories. That's the second expectation. That we're going to call an escape average fitness for the antibody. So if we now compare how, and the antibodies optimized with this framework, we're going to refer to them as shapers. And this is, and furthermore, we'll call them, let's say, horizon 100 shapers, if the horizon length that we modeled for the viral escape is 100 steps. So in this plot, this is our first result. So we're moving on to the results section, and we'll show you the results of our experiments now. And all of these results are done with dengue virus, the dengue viral antigen as the antigen that we're optimizing against. So here in this plot, what we're showing is shapers in orange, and they are optimizing with horizon 100 steps, and myopic antibodies in blue. And then what we are comparing on these axes is at the bottom, the x-axis is the myopic antibody fitness. So if the antibody was just optimizing directly for this, but just for the starting virus, versus this escape average objective that actually accounts for the full 100 steps of the viral escape. That's the y-axis here. Was there a question?
No.
Okay.
Someone I muted.
Yes, so, okay, perfect. So what you can see here is that if you look at the shapers and if you look at the y-axis, the top percent of the shapers do better than any of the myopic antibodies on the escape averaged objective. But you can probably also see very clearly that there is a trade-off, and that is that the myopic antibodies, as you would expect, do much better on the myopic objective, so the x-axis. And you can see it more clearly here. So this is like a sort of, from ML, this would be like a classic learning curve, where the x-axis is the viral escape steps. So these are the steps in the loop on the right-hand side of our like two loop diagram, or the inner loop in the meta-learning terms. And then on the y-axis, you have the viral fitness. So from our perspective of like the optimizers of the antibody, we want the y-axis score to be low. Low is good for us. And then in solid lines, you can see the performance of the shaper antibodies. So a given antibody is already fixed. And what we're showing you is how the viral escape changes as the virus escapes. And then in dashed lines, you can see the myopic antibodies. And the different colors correspond to different stages of the antibody optimization. So the fully optimized antibodies are in the darkest purple color. And now if you compare the dashed lines to the solid lines, you can see that the myopic antibodies, the viral fitness is low at the beginning. So the myopic antibodies do very well at the beginning of the viral escape, but then the virus, as it optimizes against this specific antibody, as it learns how to escape, it manages to escape them pretty well. So the viral fitness on these dashed lines goes up quite quickly. Versus if you compare it to the behavior of the shapers, so the solid lines, you can see that they sacrifice some of their performance at the beginning. So they start off with a much higher viral fitness, but then the virus can't escape as much. Does that make sense? Are there any questions?
Yeah, sure. I'm still observing the images. I don't know about others, but yeah, it makes sense.
Okay, cool. Yeah, please feel free to jump in at any point. And then we can look at this from a different perspective, from the perspective of the outer loop. So, is there a question in the chat?
Yes.
I think Sebastian will get it for you.
Yes. So the question is, different selection pressures induce evolution of different strains of virus. How are you able to mimic the conditions that can produce the same strains of virus as those that can be produced when the virus is in the body? And how confident are you that those strains of virus will be produced? So I'll break it down into like these 2 points. So I think the question is, how confident are you that different viruses can be induced? And in our model, this is one of our results. We show that the viruses that are induced, as this is showing different viruses are induced, it's not just the antibody. And we go further to show the viruses are their qualitative differences. It's not just their binding strength of the antibody. It's not just that these antibodies are better. The viruses actually appear different in a sense. I think your second point is asking about real world conditions. I'll say that we have a relatively low confidence in like anything to do with the real world. Absolut as a model, we really picked it because it was very fast, but it doesn't capture a lot of things that are very, very important about the real world. And so because of that, the idea that we would not actually think that these antibodies, if they were to be deployed, would be more effective if they were effective at all. So this is all just a simulation as a first step in order to identify these methods work. What we're interested in is like taking these ideas and then deploying them to much more robust simulations where we have a much higher confidence that these antibodies would be realistic or that these viruses would be realistic. Does that answer your question?
This was innocent. I don't know.
Okay. Sure.
But maybe if it's not certain, maybe you will give a follow-up question.
Perfect.
Yeah, feel free to write a little follow-up. All right, I'll keep going for now, but if there are any questions, jump in.
Maybe for me, I had, when you go back to the previous slide, yes, this one. Between the myopic shapers and the shapers that are within the top 10% or the successful, I'm wondering about what brings this classification I'm seeing on this plot. Like, is it from the process of optimization of these antigens or it is the same optimization, but we end up with different shapers with different capabilities? I'm asking why others are within the top 10 and it is a random process, something.
I see. Okay. So every single dot on this scatter plot corresponds to a single antibody that was generated through the optimization process. Now, the shapers were generated in a way where when you simulate the escape of the virus in this antibody performance evaluation step, you're going to simulate it for a horizon 100 escape steps. So the antibody sort of like optimizes against a virus that will be escaping for 100 steps. Versus the myopic antibody never see the virus change. So the myopic antibody only optimizes for the objective of binding to the starting virus. It never sees any changes in the virus. And then, so that's what gives us shapers and myopic. And then we can create many different antibodies in that way. And every single dot in that scatter plot is corresponding to one of the antibodies that were either optimized as like 100 horizon 100 shapers or the myopic antibodies. So that's how you get these distributions. And then the top 10%, which is actually what you asked about. So this is just with respect to these objectives. So this is if you order the shapers distribution according to their score against this y-axis objective, so the escape average fitness, you can pick the top 10% of those. And that's what this cutoff line is showing. So it's just the top 10% highest scoring ones. And similarly for the green line, it's the top 10% myopic antibodies that score the best according to their myopic objective. So what this represents is basically if you were to optimize antibodies myopically and then you would pick with respect to the objective that you're optimizing for. This is what you would pick. Those would be like your best antibodies. And similarly for the shapers, this is like a true objective sort of that you're optimizing, the y-axis. The top 10% is likely the ones that you would be selecting out of. So this is what these correspond to. I don't know if you wanted to add something.
Just to add a bit more context, the reason why there's a wide distribution of points is because the way this optimization starts is we start with random antibodies, like literally random, every amino acid is random. And because of that, the optimization process is different. There's also a bit of randomness in that the mutations you sample are random, and there's a little bit of randomness that comes from the fact that for the shapers, the shaper objective is an expectation. So it can always be a little bit different. Like an antibody that might have been on top due to randomness might not look as good. And so from those sources of randomness, you get these like different things. And there's also a lot of like, I think all of these antibodies are going to be more or less like locally optimal. Like these things stagnate. So these are all antibodies where if you make any change to them, they'll probably end up doing worse at their task. So just give more context to why there is this spread, this distribution.
That's great. That's great. Thank you.
Yeah. Does that answer your question? Is that clear enough?
Absolutely. Yeah.
Oh, great. Perfect. All right. Any other questions?
So far, no, I think.
So now we can compare antibody shapers optimized with varying horizons. So now we'll, like before, we just had myopic and shapers, and the shapers were optimized with horizon 100. Here we'll show you a spectrum. So we go from myopic to horizon 5 shapers, horizon 10, 20, and 100. And those are indicated by different colors. These are results of experiments repeated over multiple different runs. And then you can see there's like a little dashed area that's showing the standard error between the experiments. But they're really quite consistent, so they almost look like just lines. What we're showing you here is on the x-axis, this time it's the antibody optimization steps. So it's the steps in the outer loop that we're showing on the x-axis. And so now the y-axis is the antibody fitness. So this time, the higher, the better. Like this is the actual sort of fitness that we're optimizing for. And this antibody fitness is always, we always show you the fitness for the 100 escape steps. And you might be wondering why we keep coming back to this 100 value. In our sort of like simulation, we assumed that 100 is a reasonable sort of like time scale that you would care about the viral changes. And this is just an assumption that we've made. So in all of our evaluations, usually when we evaluate, this will be against this like horizon 100 objective. All right, so in terms of what you can actually see in this graph, what this is showing you is that the closer to horizon 100 you are in terms of like your shaping objective, or the objective for the antibody shapers, the better you do, which is what you would expect. Like you would expect the antibody fitness to be higher, the sort of like the more thorough you are in anticipating the viral escape and for how much longer you anticipated to be escaping. So this result is like validating our approach in a way, but this is also something that we would very much expect. However, this result is also hiding a certain important fact, and this other result shows this better, and that is that meta-optimization is computationally expensive. So if instead of on the x-axis, if instead of showing you antibody optimization steps, we actually show you how many times we had to query our binding simulators or how many samples we actually had to take, you can see that the optimization where the horizon is longer, it takes more overall steps to achieve certain results. So if you had a fixed computational budget, which you can imagine if you use a much more expensive simulator, or maybe even you actually run experimental results, like you run experiments in an actual lab, obviously like your time scale will be like bound to, like you have like weeks or months or something to deliver certain results. So feasibly, there's only that many times that you can try to evaluate this binding. So what this other result is showing is that the optimal optimization horizon is dependent on the computational budget that you have available. So eventually, the Horizon 100 shapers will do the best, right, out of all of these that we show you. But if you have a fixed computational budget, it's something that you might want to take into account. Is there something you want to add?
No, I think that was a good explanation.
Okay. All right. Any questions?
No, not yet.
So now we move on to, I think, Seb's favorite result. And also a slide title. That's his favorite. And that is a first sort of result that will be in this like realm of more explainability of what actually happens. Because so far what we've shown is that if you take into account this more long-term objective, the fitness objective for the antibody, then long-term shapers do better. But we don't know what they do to do better. And we hypothesized that there's sort of like two different strategies that they could deploy. And that is a good defense strategy and a good attack strategy. So they could either become very robust, and that means however the virus evolves, like whatever changes in the virus happen, they will like be robust to these changes. So they will maintain decent binding no matter what, or a decent like payoff. Or there's a different approach, which we would call an attack, which is they don't just become robust, but they actually actively shape how the virus evolves. So they change the viral behavior. And we wanted to see which one of these the shapers do to do better than the more myopic antibodies. So we did this experiment that maybe takes a little second to wrap your head around. So we generated antibodies for each of these horizons that we also had on the previous slide. So myopic, 5, 10, 20, 100. And then we simulated the viral escape to those antibodies, and we saved the viruses that were induced by these antibodies. So say that we had like antibody shapers optimized with Horizon 100, then we're going to simulate the viral escape to those antibodies, and we're going to save these antibodies. Those are our V100 antibodies now. Sorry, the viruses, V100 viruses on the x-axis. So the x-axis is showing you viruses induced by antibodies with a certain horizon, and the y-axis is showing you just the antibodies optimized with different horizons. And then we sort of like do this cross evaluation, hence why this matrix, where we evaluate the payoffs of like any antibody to any virus induced by a given antibody. And then what you can see very clearly from this plot is that for the V100 column, there is this, the values there are higher. There's like a much darker color in there. And what this means is that the viruses induced by the Horizon 100 shapers, they don't just, the antibodies didn't just become robust to them. The virus actually became more exploitable. So these viruses induced by the 100 shapers, the Horizon 100 shapers, they are more exploitable by any antibody. And in fact, they're more exploitable by the more myopic antibodies. So you can see the darkest squares, or like A5 V100, so and A10 V100. So these like antibodies optimized with a horizon length of 5 or 10 exploit these viruses that were induced by the Horizon 100 shapers a lot.
Alexander,
Mhm.
about this very one, it's quite exciting. The viruses that have been induced by the antibodies like the A100, and would it be able to say that the antibodies have influenced the mutations of these viruses?
Yes, I think this is what this result is showing. So it didn't influence necessarily how much the virus mutates, but it influenced the way that the virus mutates.
Yeah, like to be clear, so we have some more results and trying to look into this a bit more in this next slides. As Alexander said, it doesn't just mean there's less mutations. The number of mutations is still actually pretty similar, but some of these mutations, they're either less effective in general, maybe they're just less harmful. But this is what I was alluding to before when I say, you know, these viruses look different, right? Like this is like somehow like an objective measure, like how do you perform against these myopic antibodies? And you can see, despite being trained for this like shaping objective, these viruses are just easier to bind to in general. So this is definitely affecting almost by definition, this is affecting the mutation of the virus.
Wow. No, thank you.
Any other questions?
No, not yet.
Okay, cool. All right. So then we wanted to understand this behavior a little bit better. We wanted to understand what makes the Horizon 100 antibody shapers, you know, like influence this viral behavior. So our first attempt at understanding this was very simple. We were just like going to look at the amino acid distribution in antibodies optimized in different ways. So here again, you can see like the antibodies all the way from myopic to Horizon 100 shapers. And you can see this trend that the Horizon 100 shapers have a sort of more uniform distribution. So all of these, you know, all of these yellow bars they approach sort of a more uniform distribution amongst all of these possible amino acids. And this is in contrast to if you compare to something like the myopic antibodies, the distribution there is much more U-shaped. And these amino acids on the x-axis here, they are ordered by their sort of like stickiness, which is the same stickiness measure that Absolut uses. So effectively our binding calculation uses. So the ones on the left side are the most sticky, and the ones on the right side are the least sticky. So the myopic antibodies started off with like having some of these like very, very sticky things and some of these very, very non-sticky things. And then with the way we
Alexander,
the shapers,
about
yeah.
About stickness, it appeared in one of the earlier slides, but I have been wondering about it. Would the one perceiving it as the bindingness and the non sort of willingness to bind, is that the right
Yes.
sort of perception?
Yes, that's exactly right.
Yeah. This also comes from like the simplicity of our model in that I think this data is just generated I don't know how they generated this data, but it's not that accurate. I think there's a lot more that goes into real world binding than this.
So yeah, so basically the way Absolut gets these scores, they're extracted from, it's quite like an old work, I think it's over 20 years old now. But they basically, at the time, they took the structures from the protein data bank, the PDB, and they assumed that each amino acid from like an antibody and each amino acid from the antigen, that they interact in pairwise ways. And then they tried to get a score, like an energy sort of a contribution score of each pair to the total binding energy. So the way Absolut calculates binding is it has this matrix, the Miyazawa Yeringan energy potential matrix. That's a 20 by 20 matrix. And it gives you a pairwise energy score between any two pair of amino acids. And and then it uses this matrix to calculate the overall energy.
I think it's this next slide actually after if you went back, I think it's like the next one after.
Yeah. We're going to talk about this a little bit in a second.
Okay.
Cool. All right. So yeah, so back to these energy scores. So just to sort of like summarize this result, this was showing that for the horizon 100 shapers, the distribution of amino acids is approaching a little bit more uniform distribution. Still, that didn't necessarily explain this behavior of like shaping of the viruses. Like it just told us the distribution was a little bit more uniform. So then we came up with this other way of trying to understand what's happening a little bit better. And that had to do more with how Absolut is constructed. So I'll try to explain a little bit how that works. So hopefully this next section will be a little bit clearer as well.
Right. I just want to also check how we have 10 more minutes, right?
Well, 10 minutes, but it's not strict like after 10 minutes we have to stop like until we are done.
Okay, perfect. Just making
Okay.
sure.
Yeah, we might
Yes.
overrun a little bit.
Yeah, it's okay.
We're approaching the end. Okay, so the way Absolut works is that it takes the structure of the viral antigen, the actual protein structure, and then it discretizes it. So it puts a blob at each amino acid. And that virus structure in this plot on the left, it's represented as these white and gray balls. And then Absolut comes up with these possible binding conformations where it takes this discretized antigen structures and it figures out where the binding region of the antibody could go within that structure. So in this case, the antibody is shown as those like red dots. And it comes up with a lot of these possible binding configurations. So there's going to be like in a magnitude of millions of these binding configurations. And these balls are just placeholders for amino acids. And then the way it actually calculates the energy score, the binding energy, is it's going to then put the amino acids from the viral sequence, which is what we operate on in our method because we make mutations directly to the sequence. So it's going to take our viral sequence, it's going to take the antibody sequence, and it's going to put it inside of sort of these balls. And then it's going to look at any two interactions that any of these amino acids in the virus are making with the antibody. So now we're going to have a binding pose that an Absolut has. It can be represented as a list of these pairwise interactions between amino acids from the antibody and amino acids from the antigen interacting together. In fact, I think it also allows for like interactions from the antibody to antibody as well, which is also something that that this calculation accounts for. But for simplicity here, we're going to ignore that. And then the way the actual energy is calculated is because you have this energy matrix that's been extracted, which is also what we use to calculate these mean energy scores on the previous slide, and that's how this energy was ordered. You're going to look up the energy score of this particular amino acid pair that you've identified as in your pose. You're going to do that for every single pose, for every single pair in your pose. You're going to sum up all of these energies, and that will give you the binding energy between your amino acids, your antibody, and your virus. So that's the docking pose. And then we thought that the docking poses can actually contain some interesting information that we can analyze for us to better understand where this shaping behavior of these long horizon shapers comes from. Because an important thing also is that the way the energy calculation is done is we're going to calculate this binding energy for every single pose that Absolut has generated. And there's millions of them. And then we're going to take a minimum over all of these poses, and we're going to assume that the minimum energy is the energy that that's the binding energy. And that is because we're assuming that the minimum energy conformation is the conformation that the antibody and the antigen would actually sort of fold into. So because we're changing the sequence of the virus and of the antibody, that also means that the minimum energy pose can change as we're making these changes to the sequences. And therefore, we thought that it might be interesting to look at these docking poses and how they change. So we introduced this notion of a pose matrix, which basically will also be a 20 by 20 matrix, where both axes correspond to different amino acids. And we're going to count how many interactions are between any of these two amino acids happen inside of the pose from Absolut, the minimum energy pose. That is a little bit involved, but hopefully that makes a little bit of sense. And now we're going to use these energy matrices to do a little bit of analysis. Oh yeah, so just to explain like how this energy matrix works. So if there's any pair of amino acids, so in this case, there's an amino acid G in the antibody and an amino acid I in the virus. If there's one interaction between them that happens inside of the pose, then that entry, so the GI entry in the energy matrix, that will be set to 1. If there were 2 interactions between these 2 amino acids in the pose, that would be set to 2. If there are 0, there would be 0. So now we can look at these average energy matrices. So we can do a bunch of runs. We can plot the energy matrices or the binding matrices for all of these runs, and then we can average over them. And this is what we're showing you here. So at the top row, you can see the binding poses, the binding pose matrices, when the virus hasn't done any escape. So the virus is just a starting virus. And then at the bottom, it's after the full viral escape happens. And then from left to right, it's the antibodies that change. So we go from myopic to the horizon 20 antibodies to the horizon 100. And these in it, like just on their own, they're not super informative. You can just see that like things sort of diffuse a little bit, but we can aggregate results from these binding pose matrices, and then we discovered something slightly more interesting. So what's the influence of the antibody shapers on these binding poses? Well, first of all, we're going to sum the antibody interaction counts. So there's the antibody amino acids on the x-axis, and there's the virus amino acids, sorry, the antibody amino acids on the y-axis, and the x-axis is the virus amino acids. So we can sum up sort of like this way, that way we're going to get the antibody interaction counts. And what we're showing you here in red and green is whether a given amino acid count decreases or increases as the virus escapes. And I want you to specifically look at this last sort of bar plot here, because this, according to Absolut, is the least sticky amino acid. And what you can see is that as the virus escapes, all of these bars there go up. So you see green. So what the virus is trying to do in terms of like its influence on the pose as it escapes, it tries to increase the number of the least sticky amino acids from the antibody in the pose. So it tries to make sure that the least sticky amino acids from the antibody go into the pose. And then if you look at the shapers, so the yellow, the page 100 shapers, so the yellow bar you can see that for the horizon 100 shapers, the virus can do it the least. So the shapers sort of like prevent this behavior. So they prevent the virus the most effectively out of all of these different antibodies from including the antibodies' low binding amino acids in the pose. And then we can look at the equivalent thing, but for the virus amino acids that go into the pose. So this is the same plot we just summed up across the other axis. And now you can see, now I want you to look specifically here, because you can see that the virus starts off with a lot of I's and M's, which are on the sticky side of the spectrum, inside of the pose. And then as it escapes, you can see a lot of red. So it tries to get rid, the virus tries to get rid of its own sticky amino acids from the pose. But you can see that the yellow bars do something quite different to the other ones. So they're the highest. So that means that the H100 shapers, they try to prevent this behavior again as much as possible. So they try to prevent the virus from removing its own sticky amino acids from the pose. So yeah, that was sort of our attempt at shedding a little bit of light on how the horizon 100 shapers influence the viral escape. These results are probably the most tricky to understand. And there's possibly better ways of presenting those, but this is the best way that we could come up with. So hopefully that made a little bit of sense. But with that, that's basically the end of the results that we wanted to show you. So we'll just conclude with the things that we've discussed. So we've introduced to you this novel framework that allows you to design antibodies using principles of opponent shaping, which is a work done previously by other people in our lab. And it's very similar to this sort of like two-wheel thing where you're trying to exert certain influence over, in our case, the virus. Then we showed that the antibody shapers can outperform myopic antibodies when we consider the longer escape horizons of the viral evolution. And then the sort of like interesting result with like attack is the best defense, which is that the long horizon shapers actually actively shape the virus into a more exploitable virus. And they don't just become more robust. And then finally, and this is very important, this is a proof of concept. So our binding simulator, the Absolut simulator, it's very simple. The way we've modeled the viral evolution, it's also very simple. These results don't have like a direct influence on therapy design as of yet, but hopefully it's a proof of concept and there's going to be follow-up work where the simulator can be improved, the way we model evolution can be improved. And yeah, with sort of like more hands on deck, we can change the way that we think about designing therapies and account for the fact that in case of viruses or also in case of, other possible diseases, that the therapies that we design, they shouldn't be myopic, and they can in fact be quite smart about how they will influence the adaptation of the pathogens. And in terms of our other story of the little picture, DALL-E failed us and we had to Photoshop the blindfold in. Yeah, so lastly, we wanted to say thank you for inviting us to present. I hope this was useful or interesting, or at least we've distracted you from doing work for an hour.
No.
And yeah, we would also like to thank our collaborators for helping us out throughout this project. It wouldn't be possible without them. And if you want to access the preprint that's currently out on archive. So you can scan the QR code and it should be there. And if you want to reach out to us, here are some ways that you can contact us. So feel free to reach out and ask questions if you want to.
Sure. Thank you. Thank you, Alexander and Kalitsi. Alexander and Sebastian, sorry. I don't know whether I should begin with a joke. Like, we were in our group on a certain platform and we were discussing this work, and somebody asked a sentimental question. You know, HIV is a big problem in especially East and Southern Africa. And someone is like, with this work, isn't HIV like done? We can predict how it is changing and mutating. As like that's just a question for the researchers. I don't know what you could say about that, but I have some other questions.
Yeah, I think this work is very interesting for that, because when people talk about viruses, they always talk about how hard the adaptation is, and this work attempts to tackle that in this way. But on the other side, the reason why it's hard to tackle for, one of the biggest oppositions with this work is that biology is very hard, and it's very, very hard to model these systems. So we're very, very interested in doing that for the next work. And I really do think what this work shows is that it's actually not that hard, it seems, to account for adaptation. Adaptation does have patterns to it. You're not just defeated by it. You don't actually have to outpace it. You can account for it. And so hopefully, with putting more work into looking at simulators, you will be able to get things that also deal with adaptation. But it's also like, on the other side, you also need to be able to produce good therapies to begin with. Like, as I sound, like HIV is like sadly very difficult to produce like vaccines for, adaptation for. And so, in a sense, the assumption of this system is you actually can tackle the base virus, but it's the adaptation that is the issue. I'm not sure how's that the case for HIV.
Yeah, I think if you had reliable ways of modeling how HIV could evolve, then this type of work could be very applicable. And also, one other thing that's maybe worth pointing out, Felipe actually brought to our attention this paper that talks about HIV specifically, and how there's this interesting behavior in HIV where like, as your immune system responds to the virus, it starts producing certain antibodies, and then these antibodies exert an evolutionary pressure on HIV. So it evolves in a certain way, and that means that other antibodies can better bind to these viruses resulting from these evolutionary pressures. And that's related to the sort of like shaping results that we show. So the like attack versus defense plot. Because maybe, and specifically for HIV, maybe it's interesting to explore these sort of like mixtures of antibodies or vaccines as a therapy, you know, or maybe you could like exert the evolutionary pressure and shape the virus into a specific way so that another antibody or another therapy could come in and catch it there, you know, where it evolved into. So, and I think, yeah, as I said, there is a line of work that looks into that for HIV. So I think specifically in this context, I think is where this work could be quite applicable, I think, for HIV.
Yeah, that was actually very interesting. Yeah, that paper was very interesting because this thing appears. So you could do that. So there's another interesting question here. Thank you. This is very good work. Isn't that adaptability a mutation of the virus affected by geographical location and temperature? Case in point is like the SARS virus, which had different mutation-based evolution pressures. Yes, I completely agree. So this work really is preliminary work. So, and I would really think that it would benefit a lot more if you could model these more things reasonably quickly. So I think things like if you were to bake this into an epidemiological model as well, that would be amazing as well. And you can imagine you sort of track where you deploy the antibodies, where you do them, and you sort of affect things there. I think that would be all great, amazing. And it's not outside of the realms of possibility, but we didn't do that in this work because we really wanted to keep it simple. And so like this is honestly all trying to go to the most basics and to prove that this is something that works. And I 100% think that these sort of ideas, you can include things like an epidemiological model. I think if I was to summarize, I think the biggest conclusion of this model of our work is that compute keeps on getting better and better and better, and it keeps accelerating. And we're at the point where you can actually do these kind of simulations now. Our computers are so fast, you can just try out all of these different evolutionary trajectories. I think a core part of that in the future, we're making sure these simulations are good and are reliable, and that they can account for these different parts of these things, integrated into this epidemiological factors as well. And I think as this sort of ideas get bigger, what we hope is that other people will be interested to produce better models that are integrated into this. And they're also fast enough as well. There's this like trade-off here that we've gone to one extreme, but it'd be nice to have more models which are quick and you can account for different things, whilst you sacrifice a bit of accuracy for a lot of speed, and because of that you can run a lot more samples. So yes, so in summary, yes, these things are affected. We would love to include that more, but it's about the balance of our times. And also we are not epidemiologists either, so it's out of our skill set.
Yeah, thank you, Sebastian. Now, I've been asking myself, why, I don't know, like why simulation, or is there a thinking about applying deep learning, like going deep learning for this work?
Yes. So originally the idea was, so before Absolut, in my head, the idea was I'll just find some good deep learning model. There'll be some model which is like learn this landscape. I just couldn't find one that was good. So usually the way these deep learning models work is that they are trained on a distribution of antibodies, but only one virus. And we need both. I think the best thing I could find was trained on like 3 different viruses and a distribution of antibodies. And I just didn't trust that model at all, basically to give anything that wasn't just total garbage. Like, to be clear, Absolut, I don't trust the real world, but why I do trust it, I trust it to give meaningful, interpretable results. I don't trust a deep learning model that is out of distribution to do that. So yeah, 100%. I think our framework, any simulation works. I think deep learning will be absolutely excellent for this. It's just no one's gone out there and like trained that model yet to put it in.
And then another way you could incorporate like deep learning would be through something like deep RL, right? Like instead of like optimizing the antibodies through like a genetic algorithm or an evolutionary algorithm like we did, you could use a different optimization strategy, and you could view like very easily, this is what our group does mainly, predominantly. Our group does reinforcement learning and deep reinforcement learning. So you could optimize, let's say, the antibodies through like RL, RL methods as well. That would be feasible too. We just thought that, like evolutionary methods are very simple. And in this case, they like yielded the results that we expected. So there was no need to sort of like do this step up of complexity. But it's also like very much within the realm of possibilities, like it shouldn't be hard to swap out the sort of like the optimization loop that we have for the antibody for an RL loop, deep RL loop.
Yeah, I mean, deep learning is just great and it's amazing, and you can do all these things. You could improve the sample efficiency. You could like actually get more automated design, because that would be another interesting thing. You use deep learning to just learn how to design the antibodies in general, and you get even more complicated when you start thinking about sequences. So you can imagine a system where you deploy some initial antibody, and then after a bit, you look at what the virus looks like now, and then you decide what antibody do I want to deploy now. And that's like a very interesting thing, because then you want to you need deep learning then, because you can't just brute force that. You really want to be able to look at the virus and determine what the best next plan of action is. But that also massively increases the complexity of the system, but in a very interesting way.
Yeah, so you can think about all these like sort of interesting transfer tasks then, because like the drawback of using, just a simple evolutionary algorithm is that whenever you make any changes to the system, you need to start from scratch. Like if you were using something like reinforcement learning or deep reinforcement learning, you would have your policy network. So that would be the, the deep learning component. And a policy gives you this like answer of like, okay, being given that I am in this state, what action should I take? And that's what the network would be encoding. So if you learn in a specific case, but then you want to transfer to a slightly different case, you could start off with that policy that you've already learned. The network would be, you would already have a good starting point, and then you would fine tune to this new, context. So for example, if you had a good policy for, let's say, one virus, and then you had a virus that's slightly similar to this original virus, you could start off from the policy that you've already learned, and then just fine tune it to find a new one. You wouldn't be starting from scratch.
Right. There's a
There is
question.
there is a question.
Oh yeah, so in case the virus mutated with another and had a binding to something complex, could this model perform to its optimum capacity? So I think so. Just to clarify, I think this question is asking what to do when you get viral crossover. So as I understand it, viruses, they can sometimes combine and join together, and then you get some parts of one virus and another virus, and it can become something more deadly. I'm going to assume that's correct. Do you think that's the reasonable interpretation over there?
Sorry,